Welcome. Today we're exploring antisense oligonucleotides, or ASOs, a fascinating class of drugs changing how we approach genetic diseases. Think of them as highly specific tools designed to interact with RNA, the molecule that carries instructions from our genes to build proteins. Unlike many traditional drugs that target the final protein products, ASOs intervene earlier, offering a chance to modify or even halt disease at a more fundamental genetic level. ASOs are short, synthetic strands of nucleic acids, custom-built to bind to specific RNA sequences within our cells. Imagine a zipper. The ASO is designed to zip up perfectly with its target RNA. Once bound, they can work in a couple of main ways. Some ASOs act like a demolition crew, flagging the target messenger RNA for destruction by a cellular enzyme called RNase H. This effectively silences the gene, preventing a harmful protein from being made. Other ASOs work by physically getting in the way, a mechanism called steric hindrance. They block the cellular machinery needed to process RNA or translate it into protein. This blocking action can be cleverly used to correct errors in how RNA instructions are assembled, a process called splicing, potentially restoring the production of a functional protein or bypassing a disease-causing segment. This ability to precisely target RNA makes ASOs incredibly promising for genetic disorders, many of which arise from single gene errors. Because they can be designed for virtually any RNA target, they offer a tailored approach to fix the underlying problem. We've already seen remarkable success stories. Nusinersen treats spinal muscular atrophy by fixing the splicing of the SMN2 gene. Several drugs like eteplirsen help manage Duchenne muscular dystrophy by promoting the production of a shorter, functional dystrophin protein. Inotersen and eplontersen reduce the harmful protein buildup in hereditary transthyretin-mediated amyloidosis. Tofersen targets the mutated SOD1 gene in some forms of ALS. And recently, olezarsen was approved for familial chylomicronemia syndrome to lower dangerously high triglycerides. Developing these therapies isn't without hurdles. Their ASOs can be quickly broken down by enzymes in the body and often struggle to get inside cells or reach specific tissues, like the brain. Scientists overcome this through clever chemistry. They modify the ASO's structure, tweaking its sugar phosphate backbone and sugar components. These changes make the ASOs tougher, helping them resist degradation, bind better to their RNA target, last longer in the body, and avoid triggering unwanted immune responses. Getting the ASOs to the right place is another challenge. Innovations include attaching molecules that act like homing signals, guiding the ASO to specific cells like liver cells, or packaging the ASOs inside tiny protective spheres like nanoparticles. For brain and spinal cord disorders, injecting the ASO directly into the cerebrospinal fluid bypasses the blood-brain barrier. The field is moving rapidly towards hyper-personalized treatments. ASOs can be designed for very rare mutations affecting only small groups or even a single individual, so-called N-of-1 therapies. This offers hope where none existed before. Research continues to explore ASOs for a wider array of conditions, including Huntington's disease and even complex diseases like Alzheimer's, where specific gene activity plays a role. As the chemistry gets smarter and the delivery methods more precise, antisense oligonucleotides are set to become a vital part of precision medicine. Their unique power to directly target the genetic basis of disease holds immense promise for treating many challenging conditions.